Welcome back. Today we will be continuing with our discussion on the semi-solid dosage forms. We had looked into the various kinds of bases which are used in the formulation of the solid dosage forms, which are of the ointment type or the paste type, jelly type, or the creams, and how each of the bases have different properties and can help in the formulation of either oil based semi-solid or a water or hydrophilic semi-solid preparation. Also, we had looked into various excipients and their roles in the formulation of these semi-solids. Today we will be continuing with the further understanding of the semi-solid dosage forms in terms of their stability as well as their manufacturing design. So, in the current lecture we will look into the basic characteristics and of the dosage forms, semi-solid dosage forms, and their pharmaceutical importance. The key physicochemical properties including viscosity, spreadability, texture, rheology and permeability. We will be analyzing the methods used for evaluating the semi-solid dosage forms such as pH, spreadability, in vitro drug release and microbiological testing. Also, the stability aspects of the semi-solid dosage forms, including the factors which affect the stability, degradation, and the pathways to enhance the stability will be looked into. So, viscosity is an important measure of the semi-solid dosage forms physical property. It is a measure of the fluid's resistance to flow and is critical, especially in the semi-solid dosage forms, as it affects the drug's release property, the spreadability, the ease of application or removal. A high viscosity formulation provides prolonged contact time but may reduce patient compliance. Consistency, it refers to the overall firmness or softness of the dosage form, which varies from liquid like gels to stiff pastes. The consistency should be optimized for ease dispensing from tubes and jars without excessive fluidity. Spreadability is the measure of how easily the formulation spreads over the skin or mucosal barriers. The formulations with good spreadability ensure uniform drug distribution over the application site, ensuring uniform release and hence the efficacy. Texture, it refers to how the semi-solid formulation feel upon application to the skin or mucosal surfaces. It can be smooth, greasy, waxy, grainy or sticky. A smooth non-sticky texture is preferred for patient comfort, while greasy textures may be used in ointments to provide occlusive effects. So, depending upon the kind of effect, the texture can be varied. Then the rheological properties, like the pseudoplasticity, refers to the shear thinning property of the solids, semi-solids, meaning they become less viscous when applied or spread. This property helps in easy application and uniform spreading of creams and gels. Hydrogels and creams show shear thinning behavior. They remain thick in the container but become fluid when applied on the skin. Thixotropy, the formulations become fluid when shaken or applied but revert to their original consistency after some time. So, we have seen all these behaviors in the rheological properties of the fluids. Thixotropy is beneficial for topical emulsions and suspensions, allowing them to spread easily but remain in place once applied. Lotions and some ointments exhibit thixotropy, helping in better retention on the application site. Elasticity means the formulation can deform under stress but return to its original shape when the stress is removed. Plasticity ensures that once the formulation is spread, it remains at the applied area in the desired shape, preventing excessive flow. Bioadhesive gels and wound dressings rely on the elasticity to maintain contact with the skin for extended periods. So, the types of flow with examples are mentioned here. Plastic flow, example for ointments. These have a high yield value. Flow starts only after applying a minimum stress. Pseudoplastic flow, the viscosity decreases with increased shear rate, shear thinning. Gels and creams, thixotropy, viscosity decreases over the time under constant shear but recovers after rest. So, lotions and emulsions are the example. Chemical properties is also important. So, the pH of the formulation should be close to the skin pH, that is between 4.5 to 6.5, or mucosal membranes to prevent irritation. A pH imbalance can cause irritation, burning, or poor drug absorption. Ophthalmic gels, for example, are to be in the pH of 7.4, which is the same as tears, to avoid eye irritation. The solubility of drug is important, and it should be soluble in the barrier, the base or the carrier, for effective drug release across the barrier. Poor solubility may cause drug precipitation, phase separation, or inconsistent drug delivery. For example, the hydrophilic drugs dissolve better in water based gels, whereas lipophilic drugs require an oil based system. Drug excipient interaction is important because excipients can either enhance or hinder the drug stability by interacting with the API. Incompatibility may lead to drug degradation, reduced efficacy, or altered release profiles. Certain, for example, the preservatives like BKC can interact with active drugs, reducing their potency. Biological properties, bioadhesiveness and mucoadhesiveness refer to the ability of the semisolid to adhere to biological tissues like skin or mucosal surfaces. Mucoadhesion is a special case where the formulation adheres to the mucosal surface, oral, vaginal, or rectal formulations. This helps to prolong drug resistance time, leading to sustained drug release and improved bioavailability. Examples are carbobol based mucoadhesive gels for buccal or vaginal drug delivery. Then the drug release and permeation further have an impact on the bioavailability of the drug. So, the semi-solid bases control the drug release, affecting how quickly or slowly the drug becomes available for absorption. Lipophilic drugs penetrate the skin faster when formulated in an oil based ointment, while hydrophilic drugs may need water soluble gels. Example, lidocaine gel topical anesthetic uses ethanol as a penetration enhancer for rapid pain relief. The drug absorption from the semi-solid formulations depends on the route of administration. For example, transdermal through the skin requires penetration enhancers like ethanol, oleic acid. Topical, that is effect only on the skin, which is local in action, remains on the surface like antifungal creams. Ophthalmic, eye administration requires sterile and isotonic formulations for comfort. Mucosal, like buccal, rectal, vaginal, these benefit from mucoadhesive polymers to improve retention. For example, the transdermal fentanyl patches release the drug slowly over 72 hours for the pain management. Occlusive effect and hydration. The semi-solid formulations form an occlusive layer on the skin. This reduces water loss, keeping the skin hydrated, and enhances the drug penetration. Example, the Vaseline ointment helps in wound healing by maintaining the moisture and preventing bacterial entry. The irritation potential and biocompatibility is important, especially for the sensitive areas like eyes and mucosa. pH adjustment and hypoallergic ingredients help minimize the irritation. Ophthalmic ointments use neutral pH and are isotonic to avoid eye discomfort. Microbial contamination is possible because the semi-solid contains water and organic material. Preservatives like parabens, BKC, or phenoxyethanol ethanol can be used to prevent contamination. Ophthalmic ointments generally contain BKC as a preservative to maintain sterility. Now, the semi-solids have to be evaluated for their various properties. The first amongst them is extrudability and spreadability. The ease with which the semi-solid come out from the tube or pack, or ease with which it spreads over the skin, is designated as the extrudability and spreadability respectively. In-house tests are available for evaluation of extrudability and spreadability of the formulation. Force required making the ointment move out or extrude through the uncapped tube placed below the plunger is a test of extrudability. So, the force with which the ointment moves out from an uncapped tube gives the force required for extruding it. In spreadability test, a small quantity of ointment is placed between two slides, one of which is movable. The force which helps in moving the slide, that is the movable slide, is and the corresponding distance to which it moves is recorded. So, the graphical plots between the applied force and distance indicate the spreadability. So, this is the apparatus for the spreadability test. In vivo permeation is required for studying skin penetration and involves a variety of diffusion cells in which animal or human skin is fastened to a holder, and the passage of compound from the epidermal surface to a fluid bath is measured. So, the human skin or a semi-permeable membrane is stretched and tied over the mouth of a funnel using glass chambers such as the France diffusion cells. The penetration rates can be quantified particularly by the radioactive measurements or by the measuring the amount of drug which has crossed the semi-permeable membrane or the tissue. The area of spread of the agent on the surface is detected with autoradiographs, allowing expression in terms of quantity per unit area per unit time. Solvents such as alcohol, water have been utilized as models chosen to have negligible solubility in the phase representing the skin, but in which the drug is fairly soluble. Chloroform and isopropyl mestarate have served as sinks. Since they are immiscible with the water alcohol, it is not necessary to introduce an artificial membrane to separate the sinks from the vehicle. The important factors influencing the release into the receptor phase are the solubility in the vehicle and the partition coefficient of the drug between the vehicle and the receptor phase. So, we can see that a membrane is spread between two chambers. The first one is the donor chamber in which the semi-solid can be spread, and the next is the receiver chamber in which the media along with the right temperature as well as pH conditions is maintained. It may be agitated, it may be thermostated, and after periodic time intervals the samples are withdrawn from the receptor chamber, which helps in analyzing the rate of the release of drug per unit time, and it can also be correlated with respect to the area. Then is the irritation test. Allergy or irritation due to specific ointment base is possible, and hence the irritancy test must be done. Irritation potential of a formulation is evaluated in vitro using the HET-CAM test on eggs. In vivo, Drey's dermal irritation test on rabbits, and in humans, Drey's Shelansky repeat insult patch test is done. These tests should be thorough and well designed. They should include human patch tests, eye irritation studies, determination of minimum lethal dose at least on 2 animal species, and chronic toxicity studies. The majority of the in vivo methods are histological techniques, use of tracers, analysis of body tissues and fluids, and elicitation of a biological response. Tissue changes in skin following the application of substances to the cutaneous surface can yield information about the specific tissue affected, so that not only absorption per se, but also the route of penetration can be found. The method is limited to dye and to a small number of other substances that yield perceivable colored end products with visible chemical reactions. The penetration of dyes, fluorescence and radioactive labeling represent the most widespread tracing methods. The studies are always combined with other techniques such as histological sectioning or chemical analysis of the tissues and fluids. While radioactive methods give information on the amount of compound that moves across the skin, they yield little or no information on the route of penetration or on the localization of the penetrant within the structure of the skin. Urine analysis is by far the most frequently used method. Although urinary method is extremely valuable, caution is indicated since the recovered agent will not necessarily be the original material or the amount needed to penetrate the skin. The use of topical agents that elicit a physiological reaction when they reach the dermis makes it possible to demonstrate not only penetration but also the time required for the reaction to occur. The method is intriguing because it is simple and has practical applications. So, the responses such as sweat secretion, vasoconstriction, vasodilation, pigmentation, vascular permeability can be recorded with reasonable accuracy by visual observation. The methods have been used for studying the in vitro and in vivo percutaneous absorption. There are three variables in all the methods: the application of the medicament apparatus and the measurement of the medicament. Stability is important. So, the physical stability ensures that the formulation retains its original texture, consistency and appearance over time. Phase separation, synergis, liquid release and hardening or caking. In a poorly stabilized cream, the oil and water phases separate, leading to reduced efficacy. The visual inspection like color change, phase separation, crystallization, centrifugation for emulsion stability, freeze-thaw cycling can be done to test the physical stability of the product. For the chemical stability, the active pharmaceutical ingredient is tested. It should not degrade over a time period due to oxidation, hydrolysis or light exposure. The API degradation, pH changes, oxidation leading to decoloration or odor formation can occur. Vitamin A is an anti-aging cream which may degrade if exposed to light and oxygen, reducing its potency. So, it is a clear-cut example of chemical destabilization of the drug. The pH measurement is done to detect changes affecting the drug stability. Assay of the active ingredient using HPLC or UV spectrophotometric techniques can be done. Oxidation testing, peroxide value for lipid containing formulations can be done. Microbiological testing can be done to ensure the product remains free from microbial contaminants during the shelf life. Fungal or bacterial growth can occur. Eye ointments and creams with high water content are prone to bacterial contamination. Total microbial count detect the bacterial and fungal contamination. PT is done to ensure preservatives effectiveness to control microbial growth, and sterility testing, especially for ophthalmic and wound care formulations, is required. Rheological stability ensures the viscosity and spreadability of the formulation over time. Increased or decreased viscosity, loss of thixotropic behavior and phase separation can occur. For example, a shear thinning gel should retain its stability to spread easily when applied. Viscosity measurement using rheometers, spreadability test, gel strength test can be used for evaluating the rheological stability. Stability against temperature and humidity has to be tested. Melting, drying out, increased water penetration or evaporation, loss of consistency can happen. Petroleum based ointments soften at high temperature, affecting the drug's release. Accelerated stability testing as per ICH guidelines, that is 40 degrees centigrade, 75% RH, can be done along with the long term stability testing conditions. Real time stability testing, that is storage at room temperature for extended periods, can be done. Freeze thaw testing to test stability under extreme conditions of temperature cycling, that is increasing and decreasing of temperature, can be done. Packaging stability ensures that the packaging material does not react with the formulation. Container leaching, diffusion of moisture, gases, tube cracking can be tested. If, for example, aluminum tubes may react with acidic formulations, leading to degradation. Compatibility testing to check interactions between packaging and formulation material. Barrier property tests, for example, moisture resistance in tubes. Leaching studies to detect migration of plasticizers into the formulation can be done. Photostability has to be done to test the degradation of the drug when exposed to light. Common issues which can be seen are the color change, potency loss and rancidity of oil. For example, retinol based creams degrade quickly when exposed to UV light. So, UV exposure testing, checking for color, odor and efficacy changes can be done. Dark storage, whereas compared to light exposed samples can be compared for the changes in the stability of the drug when exposed to light. Shelf life determination is the time period when the semi-solid formulation maintains its intended physical, chemical, microbiological and therapeutic properties when stored under recommended conditions is there. So, factors which can influence the stability and shelf life of the semi-solid dosage form are like temperature. High temperature can accelerate API degradation and base destabilization can occur. Humidity, high moisture can promote microbial growth and hydrolysis. Change of pH can lead to degradation of API in acidic or alkaline conditions. Oxidation, especially of lipophilic drugs and excipients like oils and vitamins can occur, leading to potency loss. Exposure to light can degrade light sensitive drugs like retinoids and antibiotics. Packaging interaction like leaching of plasticizers or diffusion of oxygen can also impact stability. So, the shelf life has to be evaluated based on these parameters. For example, a topical antibacterial ointment stored at 40 degrees 75% RH shows 5% degradation in 6 months. So, it can be used as technique for studying the shelf life for other types of semi-solid dosage forms as well. Final shelf life assignment, less than 6% degradation can lead to 2 to 3 years of shelf life, 10 to 15% degradation up to 1 year of shelf life, more than 20% degradation, formulation, reformulation is needed. So, the accelerated stability testing is carried out at high temperature and humidity to speed degradation and predict long term stability. Data is recorded and is used to estimate the real time shelf life using RNS equation. The common conditions like 40 degree centigrade 75% RH for 6 months or 30 degree centigrade and 65% RH for 12 months can be used for finding the rate of degradation of the drug in the semi-solid dosage form. High performance liquid chromatography or UV spectrophotometry can be used to quantify the drug concentration and the changes over a period of time, and both real time, that is for freshly prepared samples, and accelerated stability tested samples can be compared to find the change in the concentration of the drug. So, the assay of the active content of the drug, pH changes which can detect the chemical destabilization, viscosity testing to check changes in consistency can be done. Unless rapid in-process methods of analysis are developed, it is a usual practice to store the semi-solids until the specified quality control tests have been completed before packaging into appropriate containers like tubes, jars or single dose packets. The product is considered to be in process until it has been packaged. These preliminary quality control tests are time consuming and delay the packaging process. Semi-solids have a tendency to set up or exhibit an increase in viscosity on storage, and such products cannot be stored for any length of time. The active ingredients in the cream or ointment may react with the storage container unless a highly resistant stainless steel is used for bulk storage. So, these issues are encountered till the product is transferred from the bulk to the individual packaging container. So, care has to be taken while determining the shelf life of the product from these points of view. Now, the semi-solids may be gravity fed if it is a 2-level operation or pumped into the filling equipment. It must be able to resist the shear stress developed in the transfer of the product as well as due to the mechanical action of the filling equipment. Once the formal manufacturing procedure has been established, there should be no deviation. If a change is necessary, the problem should be carefully re-evaluated first in the research and development laboratory and then at the pilot scale. Cleaning of the large scale equipment is facilitated, and labor costs and downtime of equipment can be reduced through use of high pressure low volume pumps The cutting force of high pressure hot water that may contain detergent can be applied like a knife edge to clean difficult to reach tight spots inside the kettles and tanks, and a variety of manufacturing process equipments can be tested, eliminating the old fashioned manual scrubbing. The packaging material used as lubricant for the shafts of mixtures should also be replaced during the cleanup process if there is any possibility that may harbor microorganisms. The pilot plant or small scale production equipment is essential in developing a manufacturing process for a production size batch. The preparation of many batches ranging in size from 2.5 to 25 or more kgs for product evaluation and clinical testing provides the opportunity to observe, correct or improve the effects of minor but important variations in the manufacturing technique or formula. Mixing and stirring operations are critical in the preparation of emulsions, and in the laboratory these operations can be carefully controlled in 0.5 to 1 kg batches of the finished product. Anhydrous ointments are manufactured by the fusion process. The active ingredient is dissolved in the melted base or wax and in one of the components of the vehicle and is then mixed with the base. The melted mass must be mixed while cooling because the fatty acids, fatty alcohols and waxes do not form true emulsions with petrolatum and mineral oil, but crystallize from the melt as temperature falls. The emulsification method can also be used for preparation of the phases. Oil soluble ingredients are weighed and placed in a beaker or container, heated gently using water bath to melt any solid components and ensure uniform mixing. The oil phase is then strained through several layers of cheesecloth to remove any foreign matter. Alternatively, the petrolatum can be passed through a filter medium, particularly for an ophthalmic preparation. The oil phase is transferred by gravity or pumped to the emulsion mixing kettle, whose walls have been heated to the temperature of oil phase to prevent high melting components from congealing. The water soluble ingredients like preservatives, hydrophilic drug are dissolved in required amount of water and also heated to the same temperature to match the oil phase temperature and prevent premature emulsification or destabilization. The components of the aqueous phase are dissolved in the purified water and filtered. The soluble drug may be added to the aqueous phase at this time, provided the high temperature does not degrade the active substance or the emulsion is not adversely affected. Otherwise, the soluble drug may be added in solution after the emulsion has been formed and cooled. The phases are usually mixed at a temperature between 70 to 72 degrees because this temperature is intimately favorable for the liquid phases. The phase mixing temperature can be lowered to a few degrees if the melting point of the fat phase is low enough to prevent premature crystallization or congealing of the components. The phase can be mixed in three ways: simultaneous blending of the phases, addition of the discontinuous phase to the continuous phase, and addition of the continuous phase to the discontinuous phase. The simultaneous blending of the phases is required to be done by use of proportionating pump or continuous mixers. The method of emulsification is satisfactory for continuous and large scale operations. The second method may be used for emulsion systems that have low volume of dispersed phase. Then the third process is preferred for the emulsion systems since the emulsions undergo an inversion of the emulsion type during the addition of continuous phase, which results in a finer dispersed phase globule. Following the addition of phases, the rate of cooling is generally slow to allow adequate mixing while the emulsion is still liquid. The temperature of the cooling medium in the kettle should be decreased gradually and at a constant rate with the mixing of the emulsion and scraping of the kettle walls to prevent formation of congealed masses of the ointment or cream, especially when the semi-solid contains a large percentage of high melting substances. Aeration may occur if the semi-solid thickness considerably upon cooling, and steps should be taken to prevent this. If perfume is to be added to an oil in water emulsion, it is best done while the mixture is at a temperature of 43 to 45 degree to avoid chilling the emulsion and facilitate dissolution of the oil. The drug is added in solution form if not already incorporated or as crystals, provided it is soluble in the external phase. An insoluble powder should be dispersed in the continuous phase prior to removing the semi-solid from the kettle for homogenization or storage. Final water volume has to be adjusted, and then the batch runs help to determine the amount of water loss on heating in the particular process, and this loss should also be adjusted. The oil film surrounding each emulsified water droplet, water in oil emulsion, tends to retard evaporation, so that water loss is not excessive following this type of emulsification. The high cost of energy has promoted re-evaluation of the processes in an attempt to limit the amount of thermal and mechanical energy required in the production of emulsions. The cost saving is achieved by heating both oil and water phase to required temperature to form a concentrated emulsion. The balance of the aqueous phase is added at room temperature during the cooling phase. Thus, the energy used to heat the aqueous phase and the mechanical energy of mixing during the cooling stage is reduced. The quality, stability, rheological properties and the particle size distribution of the internal phase of the finished product prepared by this methods depends on variables like the temperature required for forming the concentrated emulsion, the ratio of the external phase to the internal phase forming the concentrated emulsion, the phase inversion temperature, the type and intensity of mixing, and the rate of addition of the external phase. Homogenization, the creams or ointments that require further treatment are then transferred or pumped to proper homogenizer, the selection of which is governed by the degree and rate of shear stress required. The choices include a low shear gear pump or a roller mill or a colloid mill, a valve type homogenizer or a suitable sonic homogenizer. Uniform dispersion for an insoluble drug in semi-solid as well as reduction of the size of fatty aggregates can be attained by the passage of the warm ointment or cream through a homogenizer or mill. Hence, to conclude, the semi-solid dosage forms offer controlled drug release, enhanced bioavailability and ease of application, making them essential in dermatology, wound healing and drug delivery. The physicochemical and biological properties of the semi-solid formulations play a crucial role in determining the efficacy, stability and patient acceptability. The systemic evaluation techniques ensure product quality, uniformity and therapeutic effectiveness. Stability studies and shelf life determination help in identifying the optimal storage conditions. Adhering to the regulatory guidelines and advanced formulation techniques improves the stability and reliability of the semi-solid products. Thank you very much for your patient hearing.